All right, thank you for the introduction and bringing all of us together for this really great talk. It's been fantastic. Um, I don't think I ever gave a talk in front of this, beautiful wheel. So that's definitely amazing. All right. My lab mainly work on experimental biophysics. In the last 2 or 3 years, we really start to feel machine learning is a great way to analyze and represent some of these data we have. So hopefully today I'm going to tell you some of the experimental work and also go into machine learning as well. So here, what I show is, we're an artist version of a person, and we know it's composed of tens of trillions of cells in each adult version of us. And we like to think each individual cell as a fundamental building block of life. And these fundamental building blocks are really physical building blocks that contain all the necessary biological information in it. And it's the adjustment in space and over time of their physical properties and their spatial temporal, coordination and interactions that together form the life with a proper structure and a function. So we're interested in understanding how we regulate the self-assembly in space and over time. So a little more motivation here, as you can see, these are two samples that we took, I think from one breast cancer patient, and even in the same breast. Those two locations are apart by 1 centimeter. That you can see in the left one is a healthy breast epithelial tissue that you can see the nice breast SNR structure with the purple as the cells and the pink are extracellular matrix. And just 1 centimeter away, this is the triple negative breast cancer. You can see that the structure is entirely messed up. Okay, and this is obviously a result of misregulation of the cells, the ECM, and also their interactions in space. So my lab are very interested in building systems, building technologies to experimentally probe the cell mechanics, ECM mechanics, and also their interactions. So for today's talk, I want to focus on this part and start with the experimental works and then go into some of the machine learning attempts that we have been doing in the last few years. So I realized that yesterday was full of cytoskeleton. I'm not going to talk about cytoskeleton today, but I do also want to mention that we actually have been working on cytoskeleton for a number of years. And one of the focus we are very interested in is called intermediate filament. Just in case, if some of you someday feel bored of, when active microscope, you should think about this. So in this perspective, we lay out some of the emergent challenge that this field needs. All right, so let's move into the 3D multicellular systems. We in general are just very interested in when I have a bunch of cells, how they self-assemble in 3D, how they form a structure, how they achieve certain functions. And in the first system I want to introduce is a breast cancer model that we grow in the lab. And simply just take one individual breast epithelial cell, MCF10, and we drop them in a mixture of a hydrogel composed of alginate and a little bit of Matrigel to provide the biology. And if we tune the gel to be softer and stiffer, the cell and allow individual MCF10 to grow in there, their performance is entirely different. For example, in the soft gel, one cell will divide and eventually grow into a hollow sphere, very much mimicking the breast SNR structure. But in the stiff gel, you can see they become entirely chaotic, really similar to what happens in the breast cancer case. So a little more detail of the stiffer gel, they don't directly go crazy at the early on. They actually grow into a solid tumor-ish ball with a filled interior filled with cells. And at about the 5th or 7th day, they suddenly starts to invade, they break their symmetry, and then go crazy afterwards. The entire process takes about 2 weeks. That you can see this invasive behavior. So if you compare, they look morphologically very similar to what happens in the breast cancer. So therefore, we claim it's a good model system to study cluster invasion, basically. Okay, since we're interested in what each individual cell is doing during this process, here is a video that we're showing. We transfected the cell nucleus to express the GFP tagged nuclear localization signal. And the video is taken by 3D confocal, but what I'm showing is a 2D projection. You can see each individual cell, and they're basically spinning in this 3D matrix and actually surfaced by a layer of the basement membrane. Here's a collaboration with the Chase group that we studied the velocity wave propagation on the surface of this sphere. And if I put the cluster back to the incubator for another 5 days, and this is what I saw, and you can immediately appreciate how crazy they become, right? It's really like a bunch of drunk drivers, so they're drive recklessly. But very interesting to look at. And in the system, since it's a 3D, we can basically track the cell migration in 3D. We can measure the cell size in 3D. And one more thing we can do is we can take an optical tweezer that my group built to go into this 3-dimensional system to measure the viscoelasticity of each individual cell at every individual time point, and basically follow what's going on throughout this 2 weeks developmental process to measure how each individual cell's mechanics evolve in space and over time. Okay, so just quickly show you a summary of what we found with the system. Here's an illustration of the cluster. We have the core on the left and the boundary on the right. And what we find is if we look at cells close to the boundary of this cluster, cell migration is faster. Cells are clearly swollen. They have a larger volume. And if you measure their stiffness, they're also softer, a lot squishier as compared to the cells in the core of the cluster, which are smaller, denser, stiffer, and also migrate slower. There's a very clear spatial pattern of the physical properties. And through a series of different perturbation experiments, we show basically this is the result of a supracellular fluid transport. So the pressure in the core of the tumor is higher, therefore it's compressed the water with ions to flow from the core to the boundary to swell cells on the outside, therefore dilute the material, make them softer. So it seemed to really reveal the important role of these supracellular fluid flow in this particular work. In another recent work, we show this supracellular or intercellular fluid flow is also important for the whole tissue mechanics, if you're interested. All right, so in the first model system, we basically show that we can go in to measure the mechanics of individual cells, and which seem to be quite interesting. It's evolving in space and time. But what we are also really interested in is what about biology? So we know mechanics change, and we claim cells are fundamental physical building blocks. They host the biology, right? But if the mechanics of the cells change, why does it matter? Does that actually change any biological functional consequence? So to study that, we use a complex system. Before I introduce, I should say, in the last couple of years, my lab has been applying different mechanical perturbations on single cells to change their mechanics, change the degree of molecular crowding in the cells. And we have shown that you can basically massage the cell enough to change signaling, cell fate, gene expression patterns, and also noises, a bunch of biological features. But the question is, what happens in a complex system? And to study that, we basically use mouse pre-implantation embryo right before they got implanted and up to a stage of about 256 cells or ish. And during this process, we ask the question how the mechanics of each individual cell evolve over time and in space, and how the transcriptomics in each individual cell evolve over time and in space. And are they really correlated? Are there causal relations of one to another? Okay. And we basically first apply our mechanical measurement tool, optical tweezer, and also imaging based. If you're interested, I can tell you more, but I didn't include the method. But you can imagine during this developmental process, we go in to map the rheology of each individual cell in space of all the cells throughout this process in the live embryos. We can basically keep watching them. And at each time point after we have done our mechanical measurement, we fix this embryo and then bring the sample to our collaborator Fitch and Vip's lab at the Broad Institute. And they are going to do an in situ RNA sequencing to check on the transcriptomics. And the way they do it is through designing a cDNA that's fluorescently tagged to basically match on a particular mRNA of interest. And they flow in the fluorescent cDNA and put it in, let it bind to the mRNA. And then we do it imaging way to basically go through the entire embryo to see where each fluorescent tag suggesting a particular mRNA locates in 3D. And each round, we can label 4 different colors targeting on 4 different mRNA. And after we finish this round, we photo bleach all of them and then do another new round. Okay. And I'll skip this part quickly. And here's one quick example of how the imaging really looks. So each fluorescent speckle suggests one of the mRNA. So they're well separated that allow us to go in to really count how many of mRNA of each specific type and where they locate. And by doing this, in the end, we can achieve a whole embryo imaging throughout this entire embryo up to about 250 cell stage. And we can have really localize this resolution to allow us to count the number of the mRNA. And we can go up to almost 150 different genes to essentially do an in situ spatial sequencing in the mouse embryo at the different stages. And here are a few examples of the embryos that we have imaged. What I show here are cell nuclei and also the cell membrane marker to allow us to segment. I'm sorry, Margaret, but this way we do have to segment the embryo to allow us to count, right? How many of the mRNA in each cell? Yes.
But can you just say a few words about noise in the measurement? Like you have like batch effects or like some batches?
Yeah. Well, what I show in the tag process is a simplified version. We actually have to tag multiple copies of the cDNA to make it bright enough. And then when you compare that with the noise, it's pretty clear. Let me continue and then we can talk more. All right. So here, basically what I show is the cell membrane segmentation, and you can see a number of the mRNA in there. And I can certainly take the number of the mRNA and starts to do UMAP or different principal analysis on the different genes. And you can see the segmentation of the different groups based on the gene expression. And the interesting parts to us is the correlation between the cell mechanical properties and the gene expression. So on the left-hand side, what I show is the spatial difference of the cell mechanical properties. And what I put here is the compliance of the cell that you can see the inner cell mass being softer compared to the cells on the boundary. And that's seems to be quite interesting and slowly emerge as the embryo develops, that you can see the cells on the boundary progressively become stiffer and stiffer, while the cells in the center become softer and softer. And the other interesting thing is that when we test the correlation between the cell stiffness and the gene expression, you can see a number of genes are positively correlated with the cell stiffness, and some are negatively correlated. Yes, Margaret.
Sorry, can I just ask, is
Yes.
your version of stiffness here, do you think it's a mass density thing, like a water content, or do you think it's like a cortical?
What we measure is the cytoplasm. I think it's likely the concentration, but certainly related to bundling of the different.
Yeah, but it could just be water content of the cells.
Yeah. So for example, here you can see stem cell related genes are positively correlated with the softer cells because those are the inner cell mass. And some of the trifactoderm genes are negatively correlated with the compliance, suggesting stiffer cells will prefer to express more of these genes. So after I saw this results, on one hand, I was really excited because cell mechanics and gene expression are very strongly correlated. But on the other hand, it's purely just correlated. So we don't see any, causal relation. I was hoping mechanics will emerge before the biology or the other way around, but I didn't really see that. So currently, we're still collaborating on perturbing mechanically the embryo, and then look at the consequence of the gene expression to see if there's something even more interesting. But as of now, I think it's quite interesting as a pure correlative results.
And this measurement is done at the single cell level?
Yeah.
So you have, and then I guess this came up before, but like the variance of the compliance across like a single state of gene expression, like.
Yeah. A great question. So, so the measurement is at different locations within one cell. That is the variance already.
I see.
So what we report here is the mere average of everything. Gene expression is simply just count the number of genes within that one cell. That's it. There's a certain level of noise. I think trying to correlate the specific gene expression with the localized compliance or stiffness will be another interesting thing to look at. Yeah. All right. So that's another experimental system that we have to study, starts to compare the mechanical performance with the biological functions. But for the remaining time, I want to jump into machine learning to tell you more about what we have done with the graph neural network to understand or predictively model some of the multicellular processes that we have been experimentally working on. And I should say the work is really mainly done by Haiqian, who's here with me today. And we can talk to you more afterwards if you're interested. I want to try to convince you graph neural network is an interesting way to start to think about multicellular systems and to store the data, and also as a way to really go back to thinking about each individual cell as a fundamental building block. So using this graph, you can start to build a cell-cell interaction of different kinds. We have been thinking about a multicellular system as a graph for quite a number of years, initially using analytical ways. In the last couple of years, it starts to do machine learning as well. So this is one of the early example that you can see what I show here is the 2D cell MDCK cells start from individual isolated cell, and then when they progressively become more and more concentrated, they form a monolayer, and eventually they suddenly just rest, and that's sometimes is referred as a jamming transition, that everything starts to stop. And the question we ask there is, can we actually identify some other parameter to help us to define different stages of this process, especially to relate that to any potential phase transition in this experimental video. And a previous attempt have been focusing on individual cell characteristics, for example, cell shape index, aspect ratio, and what we're thinking of here is, can we do a more continuum way? Okay, and as you can see here, so what we did is we have nuclei of each individual cell, and we locate the center of the nucleus, and basically use the nuclear location to triangulate this entire space. And that's similar to a graph idea to treat this 2D space. And you can do this in 3D as well. And for each of the triangle, you can define their coordinate. And by doing that, if I compare one time point and the next time point, or 10 time points later, I can start to ask what is the deformation gradient of each triangle. And specifically, we separated two parameters there. One is how much area change of each triangle is a swelling or shrinking by calculating this determinant of this deformation gradient. And the other one, we call it a shear, is how much distortion each triangle is if I compare and use the equilateral triangle as the reference state. And here, each point is representing a single cell in this on this frame. You can see, they're really dispersed everywhere. And by using this dispersion of the cloud, we sort of call this a configurational fingerprint. But what's more interesting is based on this distribution of all these points, you can even start to define other parameter. One we use is the variance of this volume parameter, and the other one is the average value of the shear parameter. So by taking and calculating both of these parameters, we can get a set of two other parameters representing this current frame. Okay, and now you can see we apply this method to look at this evolving, cell monolayer at a different time point. And initially, both of these other parameters are high, and they both are decreasing over time as the cell density increase. And at some point, the volume other parameters stop changing first. That's actually indicates the time point where the monolayer, the cells become a monolayer, that they're confluent. Therefore, they don't swell and shrink too much as compared to their individual non-confluent state. And the other time point is later on at an even higher concentrate density of the cells, the shear order parameter stop changing as well. That suggests the cell pack now have a certain rigidity to fight against the distortion, right? So that's more like a fluid to solid transition or jamming transition, if you will. And we can apply the set of analytical order parameter calculation to a different system. Here is fruit fly embryo development process. And you can see there's a sudden transition in both of these parameters when the imagination starts to happen. So clearly, this set of other parameter based on the translation can tell us something about the order of a living system on 2D. But as I mentioned, you can do this in 3D as well. So there are a number of interesting follow-up works based on this other parameter. One work is from Karen Keza's group at Columbia. They apply this further in the embryo, Drosophila embryo, and they even show during different mutation, this set of other parameter can tell you, where the stages starts to become different upon a certain mutation. And here's another work from O.V. Charter's lab applying the set of other parameter in breast cancer. They can show the invasive tip seem to be really different as compared to the other part of the tumor system. All right. So what I showed you are 2 or 3 selected experimental systems that we have. But my students have been working on a number of other systems, and they really have been generating a lot of data. Sometimes we generate terabytes of data per day. So you can imagine it's a nightmare to look at, to think about what analytical way that we can use to understand the system. So from that perspective, I think machine learning is a pretty natural that it came into our mind that we're thinking, what can we do with it? With this, the help of the machine learning, can we start to pipeline some of these processes to have a much higher throughput analysis to help us understand what's going on in the system, okay? All right, so a little introduction of the model that we're using. So we have heard some about the pixel-based CNN type of model, and we have also heard the graph-based model yesterday and today as well. And what I want to hopefully convince you is when we think about the tissue is built of many, many cells, you can essentially think about each cell as a node on the graph neural network. And then the edge basically represents cell-cell interactions, right? So it's a very natural way, at least in my mind and in our mind, that you can think about living systems as the cells on the node and interactions on the edges. You can have different cell properties, their stiffness, their biological functions. You can have the interactions of different kinds built in the edges. So you probably don't need a more introduction of the graph neural network. But just for fun, I want to show you here as a graph representation of the Harry Potter chapter of secrets, that each character has, their features on the node, and different interactions are, on the edges of this, right? So it's a pretty natural way to think about a very chaotic, complex dynamic system composed of individuals. And one more benefit I want to say is in the field of tissue morphogenesis, there have been two classic ways to think about the living system. So one is colloidal perspective. As you can see, this is one of our own work that studies cell migration and packing on a curved surface. We basically treat each cell as a colloidal particle, right? And some early work from Dave Wade's lab and Jeff Fredberg claiming this process even behaves as the colloidal glass in some of the perspectives. And the other classic view is the foam view, that if you simply ignore cell nucleus, you only look at the cell edges, they very much look like a foam, right? And all of these are cell membranes. And this can did it more than 100 years ago, and some recent work here. And using a graph representation, essentially, we can actually combine both of these views. For example, we can have one graph based on cells as a node and interactions between cells on the edges. And we can have a separated graph, simply have the cell boundary at the edge. And then each node now is simply a vertex here on this foam wheel, right? So by combining them into a dual graph, we essentially evolve all these informations into the learning and predicting process. So just quickly tell you what we have done. This is one example that we start to ask this question. So here again, as you can see, it's a 2D monolayer formation and development process. The question we ask is, if I simply just take one snapshot, can I predict how quickly cells are migrating, right? Just from a snapshot. And we use the dual graph idea, and here is some of the details of the training and the modeling. And for example, we consider cell area in the perimeter and others as the node features. And we have the edge length and also direction as the edge features of different kinds as an example. And here's a quick results that you can see. The y-axis here is the model prediction results of the cell migration speed. And the x-axis is the experimental truth that we directly measured, right? And you can see the prediction agrees really well with the ground truth of the experiments. And if you compare that result with what people have been doing in the field, for example, compare the cell migration speed with the shape index, which is also correlated, but the correlation is not nearly as good as what the neural network can do. So at this point, when I saw that Haiqian showed me the data, I was fully convinced that this is a great idea. It's really cool. Before that, I was very skeptical about this. Anyway, but what about the physics, right? It's always a question. It can do amazing work, but I like to understand what's going on. So we are currently doing some attempts, and one of the attempts being this ablation study. Essentially, when we train the neural network, we put in different, parameters onto the node and edges, and you can selectively ablate some of these information from your model, right? Let's say I hide the edge information and see how the model does, and you can see the performance basically reduced. And you can also hide area or perimeter, and you can see the performance also reduced. That basically tells us all the information that we ablated from the model is important, right? We don't know quantitatively how exactly these parameters are involved in the prediction, but at least we know they're important. So, we're doing some other works trying to reveal the potential physical reasoning behind this. But if some of you have better ideas, I would love to hear more and talk to you about this. All right, so the next task we asked is, can we do better? So for example, you have all seen this beautiful video multiple times. This is a fruit fly embryo development, early embryo development process. And each fluorescent dot labels a cell. You can see it's very fascinating and very rapid dynamics. The cells are migrating, they fold into 3D, form interesting patterns. And the question we have is, can we use a graph neural network to predict what each individual cell is doing during this process, okay? And the first question Okay, so here's the data. For this one, we're collaborating with Homer Stern's lab at U Michigan, who has taken full embryo 3D video with laser light sheet and fully segmented that you have a beautiful 3D embryo that we can play with. The first task that we ask is, can we predict the cell migration speed? Can we predict a junctional loss in a cell-cell rearrangement? So again, we're using this dual graph idea containing both the cell position and the cell boundary position. And here are some details of the model. Here are the prediction. So to show you more clearly with the video, so what I'm showing you here is 4 different views of the same embryo, because the 3D embryo that we take front view, back view, top view, and the bottom view. Okay, and what I show here is the black dots labels the junctional loss between a pair of cells 1 minute in the future that we're that label from the experimental data set. And the colors are labeling our model prediction. So the blue color basically suggests the cells that we believe is intact. They don't lose their junction 1 minute in the future. And the pink color labels the cells that the model predicted that will lose the junction between them in 1 minute in the future. And if you look at the video, you can see actually the black dots and the pink cells actually overlap fairly well. It works even better on this very slow projector, since on my screen everything is just flashing so quickly. But here you actually see in the video slides. But you can immediately appreciate the overlap is fairly well, actually, right? And to highlight a few frames that we can see even better, here are again at one particular time point. I'm showing you the 4 views that you can very clearly see the ground truth labeled in black here overlap with the pink cell fairly well in this entire embryo. And we can achieve accuracy of about 90%, to predict what each pair of cells will actually do in terms of their junction in the next minute. And you can also do a Bayesian study to see what parameters are more important. Again, here we show the cell area, their perimeter, and their edge information are all contributing to this model prediction. I would really love to know better what exactly is happening in the prediction. So again, if you have some idea more and think more about this. Sorry. All right. Okay. So the next task we ask is, can we do even better? So the last task, what I show is what happens to a pair of cells 1 minute in the future, right? So here, what I want to ask is, can I even predict when a certain event will happen, right? Again, in this embryo, what I'm labeling here are 3 types of different scenarios. One is imagination, which means the cell on this embryo initially is on the surface. After a certain amount of time, it starts to fold into 3D. And what I label by color here is a specific time that it takes for each cell to fold into 3D, okay? And here is a time until division, which basically is how much time it takes for each cell to divide. And here is another one, is a T1 transition, basically when a pair of cells will swap neighbor to lose their junction. And again, the color label their time. So what we're interested in this task is, can we predict each of this behavior, in particularly when each of these behavior will happen in future. Again, these are the model details. And here we split the task into 3 halves for each of these specific prediction tasks. Just quickly show you the results. So what I'm plotting here is y-axis is the prediction, x-axis is the ground truth measured from experiments for each of these behaviors. And you can see that they fairly agree well with the ground truth for each of this particular type of movement that will happen to the cell. And right now, it really starts to ask when it's going to happen, right? You know, is it 1 minute after now, or 10 minutes after now, or half an hour after now? So it seemed to be able to do a fairly good prediction at this point. Again, I think I was really mind blown by you know, seeing how nicely this machine learning approach can do. I would love to understand a little more, but still, you know, the performance itself is really cool, I think. All right, so just tell you a little more of what we're currently doing. In this case, again, we're taking this full embryo, and we have our learned model. We start to pull parameters from the learned model and do UMAP to project them. You can see different part of the embryo cells starts to segregate into different groups again, suggesting the parameters used to learn that particular region is different from some other regions, right? So hopefully that can also lead to some mechanistic understanding, which we currently are exploring. And the other thing we're doing is in collaboration with at Martin's lab, that we start to play with the mutant embryos in a large quantity, in hundreds of them, to start to see if using this, we can start to learn how each type of mutant is perturbing the signaling pathway or different specific rules that regulate the developmental process. All right, so for the time, let me just end with some interesting vision, especially after seeing Susa's talk before lunch. So, you know, in the last a few decades, there have been a lot of interesting work on the protein, not only the model development, but the huge data bank that support the discovery of this protein-based model. And in the last couple of years, people have been spending a lot of effort on understanding individual cell. Virtual cell is a great example. There are different types of omics, transcriptomics, proteomics, and also great model development aiming to build a foundation model for virtual cell. And what about this multi-cell field? I think it's a great and a fantastic opportunity. It's really great to see the Allen Institute is you know, pushing forward toward this direction. It's great. And in particular, I think the model development and the database building needs to go hand in hand in pushing the development in this direction. And as an example, I have shown you the number of different experimental systems we have, and we're currently working on sharing the large scale amount of these data in some way. But I really believe this needs to be done in a collaborative way. So, you know, we collaborate using different ways, different methods, different machine learning method, and also different data acquisition, trying to see what will really make sense to push forward in this field, I think. With that, I just want to thank my students, collaborators, and the funding support. In particular, I want to thank the biggest guy here, Hai Chen, who's sitting right there. You know, he's the main driver for most of the stuff I talked today. And he's on the job market this year. So, you know, if you happen to have a good opening, he's great. All right, thank you very much.
Questions?
Hi, real question. So, if we try to imagine the metric that you measured with the energy of rearrangements, is it at the end? Is it about cell tension in the mechanistic sense? Is it the cell tensions that govern the parameters that you're measuring?
Could be, yeah. The tension, I think, could be. And normally, you know, for example, in the embryo, people have been claiming the tension correlates with the length of the cell edge. So we have the cell edge length as one of the parameter in there. It could be tension. It could be, you know, the amount of protein on the cell edge also. I don't think we know in particular, yeah, but I think it's very possible. But I should say, if you purely just based on the tension, you model this behavior, the performance is not as good as what the GNN can do. So it's more than just tension.
Right, but part of the vertex models are just looking at cell shape.
Yeah.
Is looking at the crystallinity or the triangulation.
Yeah.
Is it a different way of looking at the vertex model?
Yeah, but the vertex model parameter is certainly in there, right? But this, it captured more than just vertex model, I think.
I'm sorry if this is super trivial. So if when the cells divide, does your graph network also change? Because how does that work in?
Yeah, well, it's an excellent question. It's an evolving graph, right? So each time frame is a graph, and then next frame is already evolved. And what's really interesting is actually the evolution from frame to frame that allows to even predict, future, right? Yes, it does change.
And do you keep the information, or is it like you just need the present data to predict the next one? Do you need the information all the way back?
A great question. So the prediction I'm showing is based on the present, but the training is using the whole embryo video sequence. The prediction in particular is based on one slice. Yeah, but I think the information was stored in there from the past, right? I think you should correct me if I'm wrong. I think got it?
I had that clarification question. So essentially you train the graph neural net on the whole movie.
Yeah.
Although you change topology of the graph frame to frame different.
That's the other thing. That's really why I want to highlight the data is a crucial thing. Thomas' data is great, but he only has 4 whole embryo.
Yeah.
So we train with 3 and the test with 1. If we have more whole embryo, I'm sure the performance will be much better. But currently, that's it.
We have time for maybe one more question.
Yeah, great talk. I'm just curious because we know that from the granular dynamics. So, for example, if you have a bunch of particle, bunch of sand, then from the same configuration, they may have very different force between them.
Yep.
Then from this cell, you use the this graphic network. So completely from the geometry, you can predict what happened in the cell motion. It's really amazing. But why you don't need the force between them?
Well, good question. I'm not sure. I don't need it. One, possibility is the force is stored in their configuration, right? For example, when they deform elastically or something, then their configuration will change. Actually, related to your question, what about biology? I think some biological information may be stored in the cell morphology as well. I think all of those are very intriguing to think about.
Let's thank you again for